Hi, everyone. My name is Maddie McLaughlin, and I am currently a fourth-year graduate student in the MSTP program at IU School of Medicine in the lab of Dr. Carmilla Evans-Molina. Today, I'm going to talk to you about how we leverage ultra-high resolution imaging techniques to quantify features of the beta cell. As a brief introduction to beta cell physiology and why we are interested in the endoplasmic reticulum, or the ER specifically, the beta cell relies heavily on calcium signaling for proper insulin secretion. Glucose-stimulated insulin secretion begins when you eat carbohydrates, for instance. Your body breaks this down into glucose, which acts as the stimulus for insulin to be secreted. Glucose is transported into the beta cell via the GLUT2 transporter, which can then undergo glycolysis or the Krebs cycle, causing an increase in the amount of ATP produced. This increases the ratio of ATP to ADP, which leads to the closure of the ATP-sensitive potassium channel. This then leads to the depolarization of the cell membrane and the opening of the voltage-dependent calcium channels. Then calcium is able to enter the beta cell from both the extracellular space as well as from intracellular calcium stores in the ER, facilitating granule release. Although this influx of calcium from the extracellular space is important for insulin release, the endoplasmic reticulum is an important store of intracellular calcium and contributes to calcium-induced calcium release and amplification of insulin secretion. Intracellular calcium is also extremely important in maintaining insulin production by serving as an essential cofactor for key molecular chaperones and foldases, maturation of insulin, and coordinating insulin secretion across the islet. Our lab is specifically interested in understanding how reductions in beta cell calcium impacts insulin production and secretion, metabolic signaling, the regulation of cell intrinsic pathways, and then how all of this impacts the pathophysiology of type 1 and type 2 diabetes. Looking to type 1 diabetes specifically, during the evolution of type 1 diabetes, a number of stress pathways in the beta cell may contribute to increased beta cell immunogenicity. Dysfunction of the ER and the development of ER stress has been shown to alter folding proteins, folding patterns, and post-translational modifications of islet proteins, creating neoantigens that are perceived by the immune system as foreign, making the beta cell more likely to be attacked. Therefore, maintaining adequate calcium signaling and intracellular calcium stores is critical to preserving beta cell health. So as I mentioned, the ER serves as the dominant source of intracellular calcium and has concentrations that are orders of magnitude higher than cytosolic calcium. To maintain these high stores, the ER utilizes the sarcoendoplasmic reticulum calcium ATPase, or the SERCA pump, shown here in yellow, to transport calcium from the cytosol into the ER. This is balanced out by the ryanodine receptors and the IP3 receptors to transport calcium from the ER back out into the cytosol. It has been shown that loss of ER calcium due to impairment in SERCA or dysregulated ER calcium leak via the ryanodine receptors leads to impairment in insulin secretion. When ER calcium levels are low due to calcium release, beta cells activate a rescue mechanism through store-operated calcium entry, or SOCE, where extracellular calcium can be transported back into the ER. In store-operated calcium entry, a decrease in the ER calcium level triggers calcium dissociation from the ER-localized calcium sensor stromal interaction molecule 1, or STIM1, which is shown in pink here, allowing it to oligomerize and translocate to the ER plasma membrane, where it binds to and activates the ORAI1 calcium channel. Calcium can then be transported from the extracellular space into the ER lumen via SERCA, leading to an increase in ER calcium. Our lab group has reported in rodents and human models of type 1 diabetes, there is not only a disruption of ER calcium signaling, but also the downregulation of SERCA2 and STIM1. Therefore, we next ask the question, how does disruption of ER calcium signaling impact organelle health? As an MSTP student at IU School of Medicine, there is a joint biomedical engineering PhD program with Purdue University. Because of this, I've had the wonderful opportunity to be co-mentored by Dr. Fong Huang at the Weldon School of Biomedical Engineering, who is an expert in the field of super resolution imaging. I have been able to leverage these ultra-high resolution techniques being developed in his lab to help us better understand the ultrastructural architecture of the beta cell using the rat insulinoma cell line model. We have a wild-type control cell that can be used to answer the question, how does maintenance of ER calcium stores and functional store-operated calcium entry impact ER structure? A STIM1 knockout insulin cell line that can be used to answer the question, how does impaired store-operated calcium entry impact ER structure? and a circa 2 knockout INS1 cell line to answer the question, how does impaired ER filling impact ER structure? Interestingly, our lab has shown that circa 2 haploinsufficient mice on an NOD background have an accelerated incidence of type 1 diabetes compared to wild-type NOD mice, suggesting the importance of ER calcium signaling in the development of type 1 diabetes. The first technique we have applied is expansion microscopy, or EXM. Expansion microscopy is able to overcome the diffraction limit of light using simple magnification. Cells are labeled with an antibody of interest. Here, because we were labeling the ER, we chose the ER protein RTN4, and you attach a chemical anchor to that antibody or structure of interest. A gel is made, and the fluorescent label is actually transferred into the gel, and then the gel is expanded through osmotic force by simply adding water. So here is an example of one of the gels I made. Here's the initial gel size. You add some water, it grows, you add some more water, and it expands even further. This technique simply separates single molecules that would otherwise be too close together to resolve. After completing the labeling process, we imaged our expanded gels using a Leica Dive confocal microscope and collected Z-stack images to ensure we imaged the entire ER volume. I import these files into Imaris for 3D rendering and analysis. This shows the pipeline that I have developed, and I will walk you through it briefly, starting with the first 3 steps. First, the image on the left here shows a 2D slice, or one of the Z slices we took on the microscope out of the entire Z stack. We were able to get the 3D rendering of the cell by putting all of those Z slices together in Imaris, and we have found it best to perform a median filter to help get rid of the noise and background and make the overall signal clearer. So here on the right, we have a 3D rendering after the median filter is applied, and it goes through all of the different stacks, and it kind of builds it up so you can see the whole cell here. And next, I just have a 3D rotation of the entire cell processed together so you can see that 3D volume. From there, we perform segmentation and we can specify criteria for detection. So we go through all the slices and make sure it's picking up only the ER structures. Then we can extrude all of those segments, and it will sum all of those individual volumes of all of the individual segments for us to give us an overall volume. So here we have the overall 3D structure. However, this time it is composed of all the individual 3D volumes that have been extruded. So the software will give us the volume, all of those individual segments, and then we can sum them together to get the overall 3D ER volume. While ER volume is an important feature to quantitate, the ER is made up of different components, including ER tubules, vesicles, and beta-pleated sheets. Therefore, we next chose the super resolution imaging method called single molecule localization microscopy, or SMLM. First off, super resolution, or this technique specifically, is able to overcome the diffraction limit of light as compared to confocal microscopy through the use of photo-switchable molecules. So here again, because we're labeling the ER, we chose to use Sec61 beta, which is again an ER protein and a nanobody. Then a laser excites a subset of those single labeled molecules. The fluorophore will turn on from this excitation, an image is collected, and then it is subsequently turned off via bleaching. This process repeats for X amount of cycles. So whereas previously single molecules very close together would be indistinguishable, because they are not all collected at the same time, but rather in cycles, all of those separate images, much like a Z-stack, can then be compiled into one image, and we can get single molecule resolution. And on the right is the ER labeled in COS7 cells in the Huang lab, applying the same technique. So here's an image of an INS1 cell that I took with the ER labeled using Sec61 beta. The gray image is the diffraction limit image, which is what you would see with classical confocal microscopy. And then the orange or red image is the super resolution image. As you can see, you can get much higher resolution using the super resolution technique. You can actually resolve key features of the ER, such as ER sheets, vesicles, and tubules. I think the tubules are especially interesting because if you look to the diffraction limited image, you might be able to tell, maybe you would think you would only have one tubule, but when we overcome that diffraction limit of light, we actually see that there are actually 4 or 5 tubules there. So even more so with the tubules, using a novel software developed in the Huang lab, you can specify a segment of the ER tubule you are interested in, and you can measure the diameter of the tubule at different points and see how it varies along the length of the tubule. So you can get this quantitative measure to compare across different experimental variables. So now that we have shown what we are able to measure with these two techniques, we can use them to answer scientific questions. So first we asked, how does ER stress, a common feature seen in type 1 diabetes, impact the ER ultrastructural architecture? So here we treated wild-type INS1 cells with either tunicamycin or left them untreated as a negative control, and we prepared the cells for expansion microscopy to look at ER volume or single molecule localization microscopy to look at ER tubule diameter. Upon expansion microscopy analysis, there were no significant changes in ER volume when we normalized to total volume between the unstressed and stressed cells. However, when we looked at ER tubule diameter, we found that there was a significant decrease in tubule diameter in the stressed cells compared to the non-stressed cells. We hypothesized this finding may be due to increased membrane curvature. Reticulon proteins are integral membrane proteins that act as curve stabilizing proteins to assist the ER in maintaining its structure. And during cellular stress specifically, the expression of these proteins increased to maintain overall ER structure, in part through a decrease in tubule diameter. Further, there was an increase in the diameter variation along the length of the ER tubules when under stress. While this has been noted qualitatively in electron microscopy images, it has never been quantitated before, further highlighting the structural changes induced by ER stress. As I described earlier, calcium plays a vital role in normal beta cell function by regulating critical steps in insulin production, maturation, and secretion. And our group has shown that levels of SERCA2 and STIM1 are decreased in islets from organ donors with diabetes, and that loss of either SERCA2 or STIM1 results in increased ER stress. However, the relationship between SERCA2, STIM1, and beta cell ER structure has not been studied. So next, we use those genetic knockout cell lines that I described earlier to compare ultrastructural changes to wild-type INS1 cells. Looking at the untreated group, there was no significant changes in the volume between the genotypes. However, when we stressed the cells, there was a significant decrease in ER volume in the SERCA2 knockout INS1 cells. We hypothesized this may be due to the cell being under an increased amount of stress and therefore activating mechanisms to reduce the protein load by decreasing protein synthesis, degrading misfolded proteins through ER-associated degradation, increasing the export of misfolded proteins through ER phagy, and sometimes, even perhaps here, reducing ER membrane size, leading to an overall decrease in ER volume. Looking to ER tubule diameter, there was a significant decrease in diameter at baseline conditions in both the SERCA2 knockout and STIM1 knockout INS1 cells, perhaps due to an increased level of stress even at baseline. Further, when tunicamycin stress was added, there was a further decrease in ER tubule diameter of the STIM1 knockout INS1 cells. Lastly, because different compartments of the ER perform different functions, we next divided the ER into two distinct regions. The perinuclear ER, which we define to be less than 1 micrometer from the nucleus border, and this is shown by the green inset, or the peripheral ER, which we define to be greater than 1 micrometer from the nucleus border, and this is the yellow inset. When we stressed the cells and looked at how the ER tubule diameter changes based on different regions, we interestingly saw a significant decrease in ER tubule diameter in the peripheral ER in STIM1 knockout INS1 cells compared to wild-type INS1 cells. This finding highlights the idea that ER tubules located in the peripheral ER, or closer to the plasma membrane, may be most impacted by STIM1 deletion, as STIM1, under healthy conditions, localizes to ORA1 channels on the plasma membrane upon ER calcium depletion and store-operated calcium entry activation. So some key takeaways from this talk. We have shown that expansion microscopy enables 3D volumetric reconstruction and measurement of the ER within the beta cell, while single molecule localization microscopy achieves resolution of ER features and quantification of ER tubule diameter in the beta cell. Both techniques have confirmed that ER stress leads to changes in ER ultrastructural architecture that we can actually quantify, which is pretty cool. Genetic models demonstrate that impaired ER calcium handling exacerbates these structural and functional alterations in the beta cell, again, quantitatively, as well as shows that there are changes in ER tubule structure based on location. And this, just to summarize what we showed quantitatively, Comparing to the wild type control cells as a baseline, when there are no stress placed on the cells, there were no changes in ER volume, but there was a significant decrease in ER tubule diameter in both the STIM1 knockout and SERCA2 knockout in SWAN cells. And when we stress those cells, again comparing to wild type as the baseline, there was a significant decrease in ER volume in SERCA2 knockout in SWAN cells and a significant decrease in ER tubule diameter in the STIM1 knockout in SWAN cells. And when we looked at the peripheral region, there was a significant decrease in tubule diameter in the STIM1 knockout in SWAN cells, suggesting the importance of STIM1 in the periphery during stop-rated calcium entry activation upon ER calcium depletion, especially in the state of stress. So with that, I'd like to thank both of my labs, the Evans-Molina lab, especially Dr. Evans-Molina, and the Huang lab up at Purdue, for really just being super enthusiastic about translating their very cool super resolution imaging techniques to the beta cell. With that, thank you for your attention.